Good morning. In this class, we'll discuss about the topic hybridization. Hybridization is a very important technique in plant breeding. It improves the genetic variability of crop plants. And what is hybridization? Hybridization is defined as the process of mating or crossing of two plants or lines of dissimilar genotype. So hybridization means crossing two parents or two plants of dissimilar genotype and producing a progeny is known as a hybridization. Now, why is hybridization done? Hybridization is done to improve the genetic variability of populations. We know that in nature or in natural or in self-pollinated population, the natural variability that is present in the self-pollinated population is exhausted gradually as a result of continuous selection. Okay, so as a result of individual plant selection, that means we are selecting the plants on certain type of morphological characters. So as a result of this, the plants or the self-pollinated population will be, the variability of the self-pollinated population will be exhausted quickly. This hybridization is an important technique by which we can improve the genetic variability of homozygous population. Okay. And now, how is this hybridization done? Hybridization it is done by crossing the pollen grains from one parent, known as the male parent, onto the stigma of the female parent. So there is a male parent and female parent. The pollen grains from the male parent is dusted onto the stigmatic surface of the female parent. And it is very essential to prevent self-pollination as well as chance cross-pollination while you carry out hybridization process. The seeds or the progeny obtained as a result of hybridization is known as a hybrid, and the progeny of the F1 or the hybrid obtained by selfing or intermating the F1 plants are called as the segregating generation. So segregating generation means the subsequent generations produced from the F1. And what is an F1? F1 is simply the hybrid produced as a result of hybridization. And the term cross is often used to denote the products of hybridization. Okay, so that is about hybridization, the definition of hybridization. Now coming to the history of hybridization. Okay, we can say that compared to selection is another method of plant breeding. We discussed this in the previous classes. So compared to selection, hybridization is it's of recent origin. In 1717, Thomas Fairchild, he produced the first artificial hybrid. It's known as Fairchild's mule, and it was produced by crossing two varieties of Dianthus, Dianthus barbatus and Dianthus caryophyllus. So by crossing Dianthus barbatus and Dianthus caryophyllus, Dianthus barbatus is called as Sweet William and Dianthus caryophyllus was named as Carnation. So Sweet William with Carnation produced the first artificial hybrid Fairchild's mule, and the plant breeder who produced this Fairchild's mule was none other than Thomas Fairchild. This Fairchild's mule is the first artificial hybrid. And the importance of hybridization. The chief objective of hybridization is to create genetic variation. We discussed that genetic variability is a prerequisite for many for plant breeding programs. The most important objective of any plant breeding program is improving the genetic variability. And the degree of genetic variability produced in the segregating generations depends on the number of heterozygous genes present in the F1 generation. See, this the number of heterozygous genes in the F1 generation in turn depends upon the genetic variability of the parental population. Okay, so parental population എത്രത്തോളം genetically variable ആണോ, genetically dissimilar ആണോ, അത്രത്തോളം more gene combinations ആണ് നമുക്ക് F1 generationൽ കിട്ടുന്നത്, or the genetic variability of the F1 generation will be improved, and as a result the segregating generations will be. Segregating generations means what? The generations produced from the F1 hybrid is known as the subsequent generations of the F1 hybrid is called as the segregating generation. So the segregating generations will be having more genetic variability. So thus it depends upon the genetic variability present in the parental population. The aim of hybridization is thus the transfer of few qualitative characters or the improvement of one or two quantitative characters or the use of F1 as hybrid varieties. അപ്പം ഹൈബ്രിഡൈസേഷൻ്റെ ഒബ്ജക്റ്റീവ്സ് എയിം എന്ന് പറയുന്നത് either transferring the qualitative traits or quantitative traits or using the hybrid, the F1 directly as a hybrid variety. Okay, now coming to the different types of hybridization. Basically, we can say there are two broad types of hybridization, that is intervarietal hybridization and distant hybridization. It's based or it is classified on the basis of taxonomic relationships of the parents involved. Okay, so intervarietal hybridization and wide hybridization or distant hybridization are the two different types of hybridization. So I repeat, based on the taxonomic relationship, that means similarity of the parents, ഈ ഹൈബ്രിഡൈസേഷൻ ടെക്നിക്ക് എന്ന് പറയുന്നത് രണ്ടായിട്ടാണ് ഡിവൈഡ് ചെയ്തിട്ടുള്ളത്. ദ ഫസ്റ്റ് കാറ്റഗറി ഇസ് ദ ഇൻ്റർവാരൈറ്റൽ ഹൈബ്രിഡൈസേഷൻ. ഇറ്റ്സ് നത്തിങ് ബട്ട് ക്രോസിങ് പാരൻസ് ഓർ ദ ടു ഡിഫറെൻറ്റ് പാരൻസ് belonging to the same species. Okay, that is intervarietal hybridization, or it is otherwise called as intraspecific hybridization. And the second category or the second type is wide hybridization or distant hybridization. We will discuss it in detail. Intervarietal hybridization. Intervarietal hybridization is a type of hybridization in which two strains, varieties or races of the same species are crossed. It is also called as intraspecific hybridization. So in intervarietal hybridization, ഏതെങ്കിലും ഒരു സ്പീഷീസിനകത്തുള്ള രണ്ട് വെറൈറ്റീസോ റേസസോ സ്ട്രെയിൻസോ ക്രോസ് ചെയ്താൽ or two different varieties of the same species is crossed, then it is called as intervarietal hybridization. It's also called as intraspecific hybridization because the two varieties are coming under the same species. Say for example, Oryza sativa variety indica is crossed with Oryza sativa variety japonica. അപ്പോൾ Oryza sativa variety indica എന്ന് പറയുന്നത്, it's indica is one variety and japonica is another variety, both coming under the same species sativa. This type of hybridization or this type of cross is called as an intervarietal cross or intervarietal hybridization program. Another example എന്ന് പറഞ്ഞാൽ two varieties of Triticum aestivum is crossed. That is an example for intervarietal hybridization. And this intervarietal hybridization or the intervarietal crosses can be of two types. It can be simple or complex depending upon the number of parents involved. Simple cross എന്ന് പറയുന്നത് it's very easy. In simple cross only two parents are crossed or two parents are involved. Example, parent A is crossed with parent B to produce a F1 hybrid. In the case of hybrid varieties, such cross is also called as a single cross. And this hybrid produced as a result of simple cross, it can be self to produce the F2, അല്ലെങ്കിൽ it can be either used in back cross breeding program. Back cross എന്ന് പറഞ്ഞാൽ it can be used as a parent, is used as one of the parent in back cross breeding program. Now coming to complex cross. So what is a complex cross? Simple cross means two parents, രണ്ട് parents മാത്രമേ ഇൻവോൾവ് ചെയ്തിട്ടുള്ളൂ. Whereas in complex cross, more than two parents are crossed to produce the hybrid. Okay, so in the case of complex cross, it's also called as convergent cross because more than two parents are involved to produce the hybrid, which is used to produce the F2. For example, A is crossed with B to produce the C, and this C is used to, or C is crossed with another parent. അപ്പോഴത്തേക്കും മോർ ദാൻ ടു പാരൻസ് ആർ ഇൻവോൾവ്, അങ്ങനെയുള്ള കേസസിനാണ് നമ്മൾ കോംപ്ലെക്സ് ക്രോസ് എന്ന് പറയുന്നത്. And it's also called as convergent cross. So complex crossing is also called as convergent cross because this program aims at converging, or it brings together genes from different parents present, or the genes present in different parents into a single hybrid. അപ്പോൾ അതുകൊണ്ട് അതിനെ convergent crosses എന്നും പറയും, കോംപ്ലെക്സ് ക്രോസസിനെ. ഓക്കെ, the second type is distant hybridization or wide hybridization, or it is also called as wide crossing. Now what is distant hybridization? Distant hybridization എന്ന് പറഞ്ഞാൽ it's a type of hybridization in which two members of the different species of the same genus or the different genera of the same family are crossed. Okay, so what is distant hybridization? Simply we can say that distant hybridization is the crossing between different species of the same genus or different genera of the same family. രണ്ട് ഡിഫറെൻറ്റ് സ്പീഷീസോ അല്ലെങ്കിൽ രണ്ട് ഡിഫറെൻറ്റ് ജീനസ്, രണ്ട് ഡിഫറെൻറ്റ് ജനീറ തമ്മിൽ ക്രോസ് ചെയ്താൽ ദാറ്റ് ഈസ് കോൾഡ് ആസ് ഡിസ്റ്റൻറ്റ് ഹൈബ്രിഡൈസേഷൻ. വെറാസ് ദ ഫസ്റ്റ് കാറ്റഗറി അല്ലെങ്കിൽ ആദ്യത്തെ നമ്മൾ പഠിച്ച ഇൻ്റർവെറൈറ്റൽ ഹൈബ്രിഡൈസേഷൻ മീൻസ് വാട്ട്, ഇറ്റ് ഈസ് കമ്മിങ്, ഇറ്റ്സ് എ ലോവർ ലെവലിലുള്ളതാണ്. ദാറ്റ് മീൻസ് ഇറ്റ് ഈസ് കമ്മിങ് അണ്ടർ ദ സ്പീഷീസ്. ഓക്കെ, സോ ഇൻ്റർവെറൈറ്റൽ ഹൈബ്രിഡൈസേഷൻ മീൻസ് ക്രോസിങ് ബിറ്റ്വീൻ ദ two different varieties of the same species, whereas wide hybridization or distant hybridization means crossing between the two different species or two different genera. So based on the type of distant hybridization, രണ്ട് സ്പീഷീസ് ആണോ അല്ലെങ്കിൽ രണ്ട് ഡിഫറെൻറ്റ് ജനീറ ആണോ എന്നതിനെ ബേസ് ചെയ്തിട്ട് it is again divided into two categories or two types. They are interspecific hybridization and intergeneric hybridization. So interspecific hybridization is the type of distant hybridization coming under the same genus. It is also called as it is coming under the same genus, it is also called as intrageneric hybridization. So interspecific hybridization is the crossing between two parents coming under the same genus, which is also called as intrageneric hybridization. For example, Oryza sativa, rice variety Oryza sativa crossed with Oryza perennis. Okay, here we are crossing two different species. Oryza sativa എന്ന് പറയുന്ന ഒരു സ്പീഷീസും perennis is another species. Okay, genus remains the same. So two different species of the same genus are crossed, and it is called as interspecific hybridization or intrageneric hybridization. And the second type is called as intergeneric hybridization. Intergeneric എന്ന് പറയുമ്പോഴത്തേക്കും രണ്ട് ഡിഫറെൻറ്റ് ജനീറ ആണ് ക്രോസ് ചെയ്യുന്നത്. സോ വളരെ ബ്രോഡ് ആയിട്ടുള്ള ജനറ്റിക് വേരിയബിലിറ്റി വളരെ കൂടുതലായിരിക്കും. ടു ഡിഫറെൻറ്റ് ജനീറ ആർ ക്രോസ്, ദാറ്റ് is called as intergeneric hybridization. In most of the crop improvement programs, the type of hybridization program we commonly prefer or we commonly carry out as intervarietal hybridization. It gives better results. Most commonly used hybridization technique എന്ന് പറയുന്നത് intervarietal hybridization ആണ്. But in some cases we carry out interspecific hybridization or distant hybridization നമ്മൾ ചെയ്യുന്നുണ്ട്. Interspecific hybridization or the distant hybridization is often used to improve the varieties of one species or for transferring desirable characters. Say for example, disease resistance. ഒരു വൈൽഡ് ഒരു വെറൈറ്റിയിൽ നിന്ന് അല്ലെങ്കിൽ വൈൽഡ് ഒരു സ്പീഷീസിൽ നിന്ന് ഒരു കൾട്ടിവേറ്റഡ് വെറൈറ്റിയിലേക്ക് നമ്മളൊരു ഒരു ക്യാരക്ടർ റെസിസ്റ്റൻസോ അല്ലെങ്കിൽ ഈൽഡോ അങ്ങനെയുള്ള ഏതെങ്കിലും ഒരു ട്രെയ്റ്റ് ട്രാൻസ്ഫർ ചെയ്യാനായിട്ട് നമ്മൾ ഡിസ്റ്റൻറ്റ് ഹൈബ്രിഡൈസേഷൻ യൂസ് ചെയ്യുന്നുണ്ട്. സോ ഡിസ്റ്റൻറ്റ് ഹൈബ്രിഡൈസേഷൻ ഇൻവോൾവ്സ് ടു ഡിഫറെൻറ്റ് ടൈപ്സ്. ഇറ്റ് ഇസ് interspecific ഉണ്ട്, intergeneric hybridization is also there. And the process by which the small amount of germplasm is transferred from one species to another is called as gene introgression. So gene introgression is a very important term. It means the genes or the germplasm, the amount of germplasm which is transferred from one species to the another. Okay, and it is based on repeated backcrossing of the hybrid progenies. So distant hybridization is nothing but crossing between the members of two different species or two different genera. If two different genera are crossed, it is called as intergeneric cross or intergeneric hybridization. And if the two the members of the different species of the same genus are crossed, it is called as interspecific hybridization എന്ന് പറയും. Otherwise it's also called as, since the two different species are coming under the same genus, it's also called as intrageneric hybridization. So when you say about this hybridization, in most of the crop improvement programs we prefer intervarietal hybridization because it's very easy and it's very convenient. We'll get fertile offsprings also in the case of intervarietal hybridization. And distant hybridization or interspecific hybridization is often carried out for specific purposes. For example, if we want to transfer a desirable character which is not available within the same species, we go for interspecific hybridization. And this distant or interspecific hybridization is often used to transfer desirable genes from wild relatives to cultivated plants. Similarly, the interspecific hybridization is often preferred in some cases vegetatively propagated species like sugarcane and potato. And this interspecific hybridization, three possibilities are there, or interspecific hybridization may give rise to three different types of hybrids. They are the fully fertile hybrid, fully sterile hybrid, and partially fertile, and it in turn depends on the chromosome homology between the parents. The parents are genetically similar, or if the parents have complete chromosome homology, that we can expect fully fertile progenies, or fully fertile hybrids can be produced in such crosses if the parents are parents exhibit complete chromosome homology. This is because chromosomes in those hybrids undergoes normal pairing at meiosis, and as a result the efferent plants will be fully fertile. Example, crosses between two different species of cotton like Gossypium hirsutum and Gossypium bar barbidans will get fully fertile offsprings. So the chance for getting fully fertile or fully sterile and partially fertile offspring depends upon the similarity between the parents. If the parents are more similar, if the parents are similar, that means the chances of getting the fertile offsprings will be higher. Okay. Now about the partially fertile cross. So what is a partially fertile cross? It is an interspecific cross obtained between those parents which differ in chromosome number but have some chromosomes in common. So that means the parents exhibit partial similarity. Similarity എന്ന് പറഞ്ഞാൽ ഇതിലാണ് സിമിലർ ആവുന്നത്? ജനറ്റിക് ആയിട്ട് ക്രോമസോം കോംപ്ലിമെൻറ്റാരിറ്റിയിൽ എടുത്ത് നോക്കിയാൽ it will be partially similar. Okay, so some chromosomes will be common to both the parents. In such cases, the FN plants are partially fertile. This condition has been reported from wheat, cotton and tobacco. In wheat, there are diploid varieties, tetraploid wheat varieties. In diploid എന്ന് പറയുമ്പോഴത്തേക്കും 2n will be 14 and in the case of tetraploid എന്ന് പറയുമ്പോഴത്തേക്കും 4x condition ആണ്. So the 2n condition will be 28. Okay, and hexaploid wheat variety എന്ന് പറയുമ്പോഴത്തേക്കും 6x condition ആണ്. x എന്ന് പറയുമ്പോഴത്തേക്കും എന്താണ്? അതിൻ്റെ ക്രോമസോം നമ്പറാണ്. 7 എന്ന് പറയുന്നതാണ് ബേസിക് ക്രോമസോം നമ്പർ. സോ വീറ്റിൻ്റെ ബേസിക് ക്രോമസോം സെറ്റ് എന്ന് പറയുന്നത് 7 ആണ്. സോ ഇൻ ഹെക്സാപ്ലോയിഡ് വീറ്റ് വെറൈറ്റീസ് 6x വീറ്റ് വെറൈറ്റീസ് ആയിരിക്കും. ദാറ്റ് മീൻസ് ദിസ് ബേസിക് ക്രോമസോം is repeated 6 times and as a result 2n is said to be 42. So in wheat, in short, we can say that wheat diploid varieties on the tetraploid wheat varieties are there, and as well as you can also find hexaploid wheat varieties. So the cross between Triticum aestivum, which is a hexaploid wheat variety, with Triticum durum, a tetraploid wheat variety, will produces partially fertile offsprings. So it is an example for a partially fertile cross. Similarly, fully sterile crosses obtained in those species or crossing between those parents which do not have any chromosome homology. So lack of chromosome homology will makes the effant plant sterile because there is no chance for crossing നടക്കാൻ crossing chromosome complementarity totally absent ആണ്. Chromosome homology is totally absent. So as a result, there won't be any normal pairing at meiosis, and as a result, lack of chromosome homology makes the effant plant sterile. And such hybrids we can also make. അങ്ങനെയുള്ള sterile ആയിട്ടുള്ള effant plants ആണെങ്കിൽ തന്നെ we can make them fertile by doubling the chromosomes through colchicine treatment. Colchicine apply ചെയ്തിട്ട് we can double the chromosome number. Example, in Nicotiana, the cross between two species of Nicotiana sylvestris and Nicotiana tumendosa. Both are 2n is equal to 24 conditionalana. നമ്മൾ ഈ Nicotiana sylvestris and Nicotiana tumendosa cross ചെയ്താൽ we'll get an F1 hybrid which is completely sterile. But if the F1 plants are treated with colchicine, we can double the chromosome number and thereby we can make a fully fertile tetraploid with a chromosome number 2n is equal to 48. Campestris. So Brassica oleracea and Brassica campestris are the two different species. So they are crossed to produce a sterile plant or interspecific hybrid which is sterile. But this sterile interspecific hybrid can be made fertile by doubling with a colchicine treatment. So applying colchicine will make the sterile F1 fully fertile. So a tetraploid Brassica napus which is fertile was obtained by crossing Brassica oleracea and Brassica campestris.